Yeah, okay. So you should be able to share now.
Yes. Can you see my screening?
Yes, yes.
So, okay, let me keep this aside. Yeah, today I'm going to just introduce the spatial transcriptomic analysis, like basic idea how what it is and what we can learn from this. So these kind of there are many questions. So even I was very much excited when I started the project. So it is very big, like data analysis wise, and to understand how the data it look like, how data generated. So that is the most exciting part for me. So that is the reason I went to the one of the core lab and I closely observed how they are generating the data from tissue to the uh, like gene expression. So then this is one of the clear picture where we can see nature methods they published on 2020. So spatially resolved transcriptomics. So where we can see the middle one is a tissue, biological tissue. So they cut the biological tissue into thin section and they place that section into glass slide. Then they transfer the uh, genes into like whatever the genes in that position, each and every spot, into another slide that is having the gene barcodes. Then they went again, they capture all these barcodes, they generate like they perform the PCR, then they perform sequencing. There are steps. So because when I look into the data first time, so I don't understand what is happening in R and why there is a image and all these things. So because of that reason, I'm explaining the introduction part today. Also, I can end of this, I can show you a demo in R using single sample. Then next, I'm going to introduce cell types and like what next session, cell types and what are the spatial cell types and what are different techniques for identify these cell types and all this. So
Okay,
this
yes.
is yeah. Next. So one of the popular data type these days is VCM data, that is 10x genomics. So where we identify, if we see here on right side, so each tissue is associated with one spot. So here it captures the spot level gene expression. So within that spot, again, you may have n number of maybe 1 to 10, 10 cells. So it can capture 1 to 10 cells gene expression. So that means each spot we have 10 cells. We don't know what kind of cells it is. So like that, we have almost 5,000 spots. Excuse me. So first spatial context is nothing but gene expression data overlaid with the tissue image. And second one is retain or like organization of this tissue and cellular microenvironment from the tissue to the spot. Then cell type identification. So within the spot, what kind of cells are there and how many cells are there. So there are, like as I said, like there are many techniques available right now, and this is a hot topic. And the 10x genomics is like very friendly, and it is very easy to use currently. So the challenge of this project is uh, place like only stay at one place, that is identification of cell types. So this is how does we get the data. So initially 10x genomics they have because why I am explaining all this is so to understand our data sets. how they are like output of data set we are going to analyze. So each tissue, like they have slides, so each slide they capture 5,000 barcodes. So they have like if you see the region, so the black region is 6.5 into 6.5. So within that region we have 5,000 barcodes. So we are going to slice the our tissue sample for suppose cancer. So ovarian cancer or tissue sample, we'll slice like slice cut. Then we can place into the one of the slide. Then we can transfer into the spots. So how each spots, like each spot is like 55 like for each spot. So each spot contain 1 to 10 cells. We can we get the spot level gene expression. Then again, each spot has the like poly like spatial barcodes. They have each spot have a number of millions of capture probes. So these probes can capture the whatever the uh, genes that is there exactly opposite into the tissue. So uh, again, next we are going to like the final step is we are going to extract this spot level like polytail. Then we are going to see what kind of transcripts are there. So here each uh, here each spot is given a barcode, right? So this barcode is like for suppose here is spatial barcode, green color. So the for suppose we have 1 to 5,000 barcodes. So if this particular barcode is a 499 number, so that means we know exactly where this particular transcript came from, like what kind of spot. So like that, we collect the all the spot level gene expression data. So from this, this is how the data generation. So next we are going to see the complete workflow. So this is how the first part is a molecular, like they call like sandwich method. So they transfer the tissue to the slide. Then in VCM there is a instrument that can transfer this process. They are different techniques, different temperature they used to maintain. So it will take half an hour to transfer the 20 minutes to transfer from tissue, like molecular sandwich method, from tissue to the VCM slide. Again, then as I said, like previous one, uh, they have barcode for each one. Then millions, they whatever they capture, they like they all are arranged in a. We can clearly see the uh, their their arrangements. Then so, as a computational biologist or bioinformatician, what we see, we will get the spatial data. So we get this whatever the slide we have, and we get the gene expressions, and we get this barcode, and all these things, and like all the associated information. So each slide, like each sample, we have like same like bulk RNA sequencing, we have TCG. So each sample we have expression, right? So here uh, each spot has expression. So people who knows the single cell, so they both are similar until the last like steps, like analysis steps, except a few changes for spatial barcode. Like single cell RNA sequencing technology is it captures the single cells, but spatial it captures the spot level expression. So this is the output file from the VCM and input file for our. So what is this? So this is the so we'll get feature like filtered feature BC matrix. So we have so within the filter feature BC matrix, we have barcodes. So barcode is nothing but each spot has barcode and features. And each spot, for suppose 1 to 100 is associated with some name. So and features, how many genes are present in each spot. So and their matrix, that is nothing but gene expression data, so present in the particular spot. So these three information we are going to connect with the different molecular information. For suppose what is the tissue came from, what is the tissue like stages, grades, and all. They are clinical parameters, right? So we are going to integrate all these clinical parameters once we have. So each sample will have this information, and apart from this, so we have two images: high resolution image and low resolution image. So and tissue positions. So these three is connecting, like tissue positions is connecting these three barcode features and matrix files together with the tissue image. So as initially I showed up, right? So 6.5 into 6.5, whatever the region it is there, exactly it merge the same it makes the layers on top of tissue. So I'll show you that in a while. So yeah, let's coming to analysis, like transcriptomic data analysis. So this is like R, we have Surat package, that is one of the great one at present. So we can we have all three data sets, like barcode, features, and matrix. And also we have different, for suppose in my project I have around 22 samples. So we are going to make each sample into like one object. So this object is nothing but a group of all the information collected together for each sample. Then the object, like, so object is nothing but yeah, so each sample we have different parameters aligned at one place. And the same way, we have 22 samples, we have 22 objects. We are going to merge all these 22 in later, like later session. So next section, I'll show you that information. Right now, I'm going to demonstrate only for single sample. Then what we are going to do, step two, is we are like removing the unwanted, like low quality spots. That is, some spots may capture maybe 10 genes expression, so that is very low. So why we are doing removing low quality spots? Sometimes if the entire spot is having 10 genes, so it may capture by like one CD4 is one of the marker for CD4 T cell. So if the expression is very high, so it is like it is considered as CD4 cell. But when it came to, we are looking into other spot, there we may have maybe 10,000 genes expression. So there their CD, along with the CD4, there is CD3D. So they are markers. So cell type specific markers. So number of markers, if the number of markers is more, the expression, like we can clearly see the spot level expression. um, then we like we remove the low quality, then we have the high quality data. So this is a normal step, even bulk RNA sequencing data. So we are normalize the gene expression data, uniform the data throughout the samples, and we identify what are the uh, highly variable, like similar expressed spots. That means if we see 10 spots like having multiple genes, so we already know like from starting of the gene single cell and spatial. So people always split the spots or single cell based on their marker gene expressions. So here uh, we don't know what is the marker. So we are considering as like 1,000 spots. So these 100 spots are highly expressed similar kind of genes. Then we used to do PCA and reduced then PCA analysis. Then we perform graph-based clustering. So then we on top of it we visualize the gene expression of tissue image. Then like we subset the regions and groups. Then we can predict the cell types. So here step 8 is the most challenging part for many in every analysis because you need to identify what kind of cell within the spots is highest proportion. Then yeah, if we have multiple sample, you need to integrate all these samples and perform all these steps. Okay, so this is I think most of the single cell analysis and spatial is similar. So graph-based clustering. So it is uh, it is cluster the similar neighbors, like similar um, expressed cells based on these spatial arrangements. For suppose if we see the some cells are very close, even some are far away. So it is capturing the based on distance. It is capturing the like which side it is belongs to. So this particular gene is belongs to this one or this one. So based on the distance, so they will uh, calculate the distance between cells and what is the closest one. So that is assigned. So there is some like this is common. This is done by developed by Lavian algorithm. So I think it is incorporated into Surat package. So you no need to get all this information usually. So this is how this single sample looks like. So first you have understand what is the how many number of spatial information is there, and number of features and percentage of mitochondria. So these three are the parameters for common parameters for each and individual. So for suppose if the percentage of mitochondria is very high, more than 20, so uh, like so there is a if mitochondrial is more, the tissue or cell is more into dying state, so apoptosis. So that is the reason one of the major step is you need to remove the cells which are in the apoptotic state. Then from so this is for suppose this is a slide from my analysis on sample. So it is like they cut this one as a round shape. So still whatever the area is captured is 6.5 into 6.5. So it may not have all 5,000 spots, but it may have 3,000 spots around. Then when we transform our data into UMAP, so each we can see each spot here, like one dot is a spot, and here is like clustering. So we have clusters, so we separate based on their expression of highly variable genes. Excuse me. So here we have 10 different spots. That means each spot is captured by one of the, so each spot is for suppose 100 people, if we can say 100 people, so 50 are from 70 are from India, and 40 are from another country. So the majority of the population, so that is what it takes. So then there is also like subcells. So it is counting how many of them are there in the spot and how many of them are there. So within the spot. Uh, yeah. Then this is how the expression look like. Uh, this is the cluster 0, so we cannot say like there is only one gene is highly expressed, IGHA2, and cluster 1 we clearly see that MMP1 and CXL. So these type of genes, CCL8. So these are marker, like we can say marker genes. So these are having biological information. So based on this biological information, we can say, okay, this cell is belongs to this. So if there are two or three in the within the spot, maybe five cells are like seven cells are CD4, as I said, and again again three cells are another. So we know how many what is the proportion, their proportion from this. So this is how we found the highly variable genes in each cluster. Then just we can see like this is for general demo. So I observe like we have 10 spots. So what is a spot is associated with CD4 cells. So when I look into the CD4 markers, CD80 and sorry CD8, yeah, CD4 and CD3 3 and CD80. So these all are CD8 cell markers. So you have many markers, not only these. Just a demonstration here. Okay. Anybody have any doubts so far?
Everybody need to enter just a minute.
Okay.
Yeah,
Okay.
Andy.
Yeah.
Andy.
So, okay. So this is the final step of what we are discussing so far. So this is final integration with single cell RNA sequencing data. So why we are integrating this information? We have marker genes from our spatial data. So we want to know each spot, each cluster is having kind of cells um, to understand that. So one approach is, so we have reference single cell data. So their markers, we are comparing their markers, and based on that we isolate the markers of cell types here. And yeah, this is the final part. So identifying cell types using different deconvolution methods. So I'm working on this one now. So I'll demonstrate this one into next part. So one is deconvolution method, most popular. So using reference based, if I have single cell samples of um, uh, spatial samples of triple negative breast cancer. So there are some standard single cell data sets that can compare to identify cell types within the spots. Okay, from by yeah, this is the
Yeah.
yeah, this is all about spatial transcriptomics. And let's see the another one. I'm just switching to the R programming right now, just to demonstrate. So this is you can also get the same information from this. Uh, a visualization and integration of spatial data sets with Surat. So you will get the whatever the data I am using from Surat. So you will get all the information from here. Uh, so what we need is we must need a good RAM for this. because small single sample it won't need much RAM, so even 12 GB is also fine. Uh, okay, let me read the okay. So now I took the information like one of the publicly available data sets, that is brain region um, of the mouse. So they have anterior and posterior region of the brain. So they collected the brain slide, and they want to identify the spatially like spatially arrangements of cell types in brain. So let's see first. So I have the data. So I want to see what is the quality of uh, okay, data layer spatial not validated. Let me restart the complete. Yeah. So whenever I tried my Sirat package for my local computer, it is always getting me a trouble because of sizes of the data. Okay, let me read this. Following will not be reinstalled. Okay, then I'm reading the brain. So load that data. So that will give me information. Yeah. So this is the brain region. They have this is within the tissue 6.5, 6.5. They are the spots. Each spot like they have number of counts for each spot, and number of features, and number of like mitochondrial information. They already have that all the information. So they I think these samples they already pre-processed that information. So this is a tissue we are going to work on. So this is a single one. Then I'm going to normalize the data. So these days, like previously, we use different 3 to 4 codes to transform the like counts into like raw rate counts into normalization. Then SAT transform is very useful. Just normalize and it will also identify variable features. Okay, so it may take very less time, maybe less than 30 seconds. Then, so next step is we are going to see the genes. For suppose, for example, in this analysis, HPCA is a strong hippocampus marker. So, TTR is a marker of uh, chor- choroid, choroid plexus. So these are the regions in the brain. So let's see. Yeah, so let's see, these are HPCA highly expressed in this region and this region. And like this is a secondary region. And uh, TTR, so highly expressed in this. This is a marker. So that is explaining us. So these spot, so here maybe a 1,000 or 2,000 cells within the spot highly associated with uh, yeah, plexus choroid plexus region. So that is what we understood from the if we can see backside is a tissue, real tissue, and front side is the like labeled one. So that is we have two layers here, and let's see next one. So these features again, mm, is happening. Yeah, I must be very small. Okay. Okay, let's do another things. Okay. So this is same like feature spot level. So I'll also explain this one in same figures. So that is giving us each spot level. So let's dive into like next one. So first one is PCA. So we have done SCT transformation in first step. The second one is PCA, and we also select the number of PCS. I can say, for suppose I want to know. Uh, like for suppose if I want to use 30 PCS, I can simply use dimensions. And yeah, PC is done. And next is find neighbors. Uh, all the like we already have the clusters. Sorry, neighbors. Then we can find the clusters. Then we usually find the clusters into UMAP format. Then it will take, yeah. Then we can see what is our UMAP look like. And okay, this is interesting. In the while, okay, something is not great new page. Okay, now I got it. Let me switch into another, yeah, because of the size and monitor size like is not enough to show up the figures. Okay, okay. So now you can see that is a problem. So yeah, sorry, my monitor size is not enough to show the figure size. So let me see. So where did we miss this one? Yeah, these are, let's, okay, this is not enough space. Then our next information is this. So now here we found clusters, uh, like neighbors marker genes and clusters. We first separate the PCS and neighbor marker genes and clusters. So now we transform this information into UMAP. So UMAP is very like good way, like nice way to understand the um, like spots. So here we can say spot level, right? So a group of spots based on their expressions. So here we'll have on right side we have 14 clusters. So that means 14 spots are their expressions are similar. So like we can see they based on their expression, so their UMAP showed up the like arrangements of these cells. Okay, spatial team plot. Okay. Now these are the, for suppose this is a one for suppose cluster one. So these are all cells are belongs to cluster one. These all cells are belongs to cluster two. So you can see all this belongs to some cells, all are having similar expressions. And let me,
Sorry, one
Yeah,
question.
yeah.
Different cluster means different cell types,
In spatial analysis,
different
different
region.
different spots. So all are
Mhm.
similar spots. For suppose, this is one of the spots. If you see, like you can see my cursor, right? So this particular cell types is having group 6. So, and that is cell type and barcode and coordinates. So based on this entire region of cells or spots having similar expressions. So again, within the spot, they are like hardly having CD4 markers, highly high expression of CD4 or epithelial cells in this marker. So in spatial, we have like downstream analysis. We need to find what is the proportion of cell types within the spots. So I can say hardly each spot, for suppose group 6, this barcode with the spot is having 10 cell types. So out of 10, 7 are highly associated with epithelial cells. So, but there are also 3 cells, or maybe they are CD4 cells, or sorry, epithelial cells or something. So that means these CD4 epithelial cells are closely associated with the CD4. So that is microenvironment level, TMC level. So we are understanding what is the proportions. Again, we need to see the proportion of cells within the first layer is these all are epithelial part. Second one is there is also a second layer. There is also second population is involved in that spots. I think, did you get my point, Professor?
Well, you go ahead first. I will raise the question after you complete.
Okay, okay, okay. So this is what if I need to just find the particular spot. So these all are associated with similar expressions, but this is, for example, this is a kind of, if we can say, one region. So within one region, each spot is having um uh, top cells with expressions, and is also second layer is always involved. So if I want study this region, so I'll just select this one. So this is a tissue and this is spot level. So let's dive into another. These are the things just we already seen this one. So each spot like parameters, like how well we want to look into visualize the intensities of expression. Always our background is tissue, and there is also one layer that is from spatial layer. And these are another example. I'll just yeah, that is saving, how we save the each. Yeah, these are these another markers which are play a key role in brain regions. So PIP1. So this is a mouse data. Yeah, and so we can see spatial expression. We can clearly see the spatial expression here. UGT. So I think there is no gene. Okay. And yeah, this is a spatial expression of ERMN gene. Yeah. So we can see some of this region is highly like expressed with these markers. And so like this is initially we have seen, right, 14 cell types. So I can just select the what kind of um, cluster I need to study in like in detail later. So if I want to focus on like outer layer, so there is these cells. I just want to click this, and I will go for next analysis. What kind of cells it is, and what is the proportion of cells it is having. Um, then this is another thing. So I want to identify the spatially variable features in, for suppose, 5 and 6 clusters, between 5 and 6. So then I just need to use like will like within this, like there is a Wilcoxon test they use. There is also different statistical tests. So to find differentially expressed markers between cluster 5 and 6. And next step is we want to see what are the top markers. For suppose this is similar to what we are doing in differential gene expression analysis between tumor and normal. And then later we found the what are the genes. For suppose CALB2 and STX1A and CAMK2 and 1. So these are highly associated with like high expression we found in outer layer, and these two are there. But CALB2 is associated with this region. So 6 and we have done between 6 and 5 clusters. Then so find spatially variable features. So they are methods. So let's yeah. So first let's see thousands features, like top 1,000 genes. Then yeah. Meanwhile, it is taking little time. So any questions, Professor, or any other?
Yes. So when you talk about the cluster, this cluster is referring to spatially close region or what?
Yes, region, spatially associated region. So this is the these all region is having similar expression. So this is a little tricky. Even I was initially very much not understanding this part. So why each spot is having, if it is having similar expression, it must have similar cell types. But later, what I understood is, so we observed based, there are many markers. So there are layers of markers. So that is the reason each spot we found the proportion. For suppose if a circle is having 100%, so 60% of one kind of marker shared the like gene expression. Like if we take 1,000 genes in the single spot, 600 genes are highly associated with one cell type, like epithelial. This is outer layer, epithelial. But there is also 400 markers that is also expressed. So, but not this much another. So that is called like neighboring. So within the spot, there are cells, neighbor cells. So they all maybe different uh, they all are kind of, you know, right, TMC is very complex. So we can see cell interactions over there. So what kind of cells, for suppose epithelial cells, they are more close to the CD4 cells in outer layer. So that is a biological thing. We if we dive into this, so that is what we can observe from this. So each spot may have 1 to 10 cells, and based on top expression, there is also second layer. So that is the reason We always single cell, it is easy. So each spot is nothing but single cell. So you can easily get all these cells are belongs to some one group of cells. But spatial, even this is closely like all these are same group, but same group of spots. So again, within the spots, you have different cells, two to three different cells. So usually we'll go for two cells or three cells down. Okay.
So let me clarify one thing.
Yeah.
So the image you show on the left, you have the blue color spots
Mm,
on the surface.
mm.
So each color, the blue color spots uh, represent a cell type,
Oh, like
referring to the right hand side, because the right hand side is the single cell cluster. So is it
no, right
the
hand
color?
side is yeah, yeah. So right hand side is spot level. So this is all our spot level right now.
For sports
So
level is not the cell type level.
yes, so that is where our next part will come. So right now we cluster these spots, similar spots with similar expression. So within the spot, again, there are cells. So that is where deconvolution methods will play a key role. So what they will do is deconvolution methods. So they will identify what proportion of cells present within the like entire that region. So um, again, this region, for suppose maybe here until here, they are for suppose these all are top epithelial cells, but this region, even epithelial cells, they are shared with other cells. Maybe as I said, example CD4. Maybe this region, this epithelial cells is shared with CD8 cells, or these epithelial cells is shared with some fibroblasts. So like that. So there is that is where we will get the cell-cell interactions and all these analysis. So that is we found in deconvolution method. That is, I think, next time I think I am going to explain in detail about the deconvolution methods. So
Oh, okay, yes, okay, then I see. Okay, yes. Now,
yeah.
in your R program,
Mm.
hey, just a minute. Uh, Now you have a command, something like find cluster,
Okay.
and you also have a command called run the UMap. So is it, are you using the Lewin or the Lewinian algorithm
Yes.
here?
So yes, so this is like these two, so they are having like within the like these codes, so they have graphical as I mentioned earlier. Um, oh, 1 minute. They use the like they have like a graph-based clustering. So they use graph-based clustering using a Liouville algorithm. So this is a inbuilt one. So we if we want we can change like there are parameters so let's simply go into this. So yeah, there are also like uh, other parameters for suppose if we want to yeah, so modularity CPM, and there are okay optimized. Yeah, so they have if we can say if we use this option, they use this algorithm and algorithm with multi-level reinforcement re uh, sorry refinement, and another one three is SLM algorithm. They have already inbuilt algorithms within that fine clusters. So most people they will use like if we don't mention that, so it will take like one mostly. So original Lovian algorithm. So if we want, we need to specify what kind of algorithm we are using here to find the clusters. So this is the similar methodology for single cell RNA sequencing. But what is the difference is image level, and at the same time deconvolution downstream, like down place we have deconvolution information to identify spot level cell proportions. Then I think did you get my point, Professor?
Yes. So for the spatially uh, the graph-based cluster, uh,
Yes.
yes, how what feature they are used to cluster something that is spatially close?
It is like one like we have find spatially variable feature. So it is right now this like at this spot it is isolated for suppose top 1,000 genes within the spots, and uh, if all top 1,000 genes are aligned with the similar expression, they set up as a group of spots. Did you get my
So
point?
to get this graph-based cluster,
Mm.
so they look for the whatever cell type, whatever the gene, as long as they have a similar expression level,
Yes,
then they put it in a using the graph-based algorithm and put it in a single cluster.
yes. So it will take for suppose in this we can see it will study the top 1,000 genes. Then it assigns because these 1,000 markers are similarly like within the one spot, similarly expressed in another spot. So they mix as a single group. And also it is consider another thing is spot, so where is this spot is associated. So uh, so these top for suppose there are two spots, right? They are away from each other, but these top spots like whatever 1,000 genes are, their expression is matching these spots. That is the reason even that is away from each other, but they consider as single equal spots. So next step is what kind of cells present in each spot, like spot level. So now we have these spots all are belongs to one group. Then they all are belong to one group, but they all are having what proportion of cell types within the spots. That is our deconvolution method will reveal us.
Uh-huh.
So as I said, like until here, there may be fibroblast plus epithelial cells. So epithelial cells is for suppose top marker. So that is considering these like all these cells, but there are layers of cells. So second layer. So we have first layer epithelial cells that makes the all the groups. So second layer up to here, there may be some kind of fibroblast or some kind of other cells, so they're mixed up. So uh, so their cell interaction, they spatially arranged. So one region is associated with epithelial cells and fibroblasts. So the cell interaction part will come once we have the uh, deconvolution information.
Oh,
Yeah.
so
So
for the deconvolution steps, this allows you to determine the fractions of different cell type and also allows you to build a cell-cell interaction network.
Yes.
Oh, okay. Oh, good.
So yeah, because that is the reason I don't want to confuse
Yeah,
at my first presentation. So
yeah, yeah.
first, they are single cell RNA sequencing is straightforward steps. So we have only single cell level expression. We get all single cell and we separate the single cells into their groups according to the marker genes. But here, the tricky part is we have like cells are arranged in each spot. So then first we identify similar spots, and second, what kind of cells or proportion of cells are present within that spot, that region. So thereby we know this region, for suppose this is a brain posterior region. So these this epithelial cells are, these all are epithelial cells, but this region is highly associated with epithelial cells with other cells. They are closely they are closely interacting each other. So we don't know, for suppose this is the region, um, particular region is interacting, so it is in disease condition. So that is what people that is what they showed. So particular region we observed interacting between these cells and epithelial cells. So there may be a biological way that there is a close uh, uh, for suppose they simply consider, so this is a tumor, these cells are closely associated with the tumor, but these are not. So what kind of cells, here if we can see, there is no CD4 cells, so there is only epithelial or some, but here we can clearly see epithelial plus CD4. So I think this information, I think next whatever maybe next week, so we can discuss about this in detail. So because I haven't completed that step yet. So that is what deconvolution will give us.
Okay, I see.
Yeah.
Okay, now back to your PowerPoint slide.
Okay.
In one of your PowerPoint slide, the
Mm.
title of the slide is single a single sample ST. Yes, this one. Um,
So yeah.
the diagram on the right hand side
Mm.
say the left one is the I, I think that is the well, you have spots with similar color, like the lower bottom of the rightmost image as
Okay.
a green. So that means those refer to the same spatial uh, region where the gene exhibit a similar gene expression. Is that
Yes.
correct?
Yes. So
Okay, okay,
uh yeah,
okay,
that is correct. So
okay.
this is I can say uh yeah, you are absolutely correct. So this is a four. Uh, this is all our spot level, not a single cell level. So this is single sample. So we have let me, so for suppose we have 22 triple negative breast cancer. So you can see these are MB1, MB2. So we have these many patients, participant patients. So when we, first thing is when we look into the single sample, so they are two things. So these single sample have these many spots and expressions. So when we integrate multiple samples, so it is also going to be the same, like where we have uh, like it will collect the similar spots for all the samples, all the 22 samples. So Then we like that is part of integration. So we'll have two U maps. This is a general U map. So this is after we perform PCA and graph-based clustering. So this is another U map. So here each spot from here. Yeah, from here. So each spot from here to here. Then again each spot we have proportion of cells. Did you
Okay,
get?
I
Yeah.
see. Yes, yes. So, so on the right hand side of yeah, the rightmost figure, you have a square with a number. So the number is referring to the figure to the left. Is that right?
Yes, yes. So let me zoom
Okay.
that. So
Okay.
so this is like cluster. So they assign as single group of cluster. So all these spots are here, like same like single cell. That is
Um,
group of single cell over there. But here group of similar spots, similar expressed spots, spot level. So then, like here is 4 transform to here, and 3 is here. So 3 is, you can see this. So UMAP will show us how these like UMAP will show us the arrangement of these clusters based on top markers and top genes. That is the reason we are observing like clear pattern. So 10 like a cell 10 like spot level cells, sorry, uh, I'm confusing. So a 10 cluster with the spots are making as a group here, but here it may be majority may be here. So sometimes we may see another cell is present another place also, sorry, another spot is present another space also. So yeah, if we can see 8, so it is like all purple color is present here. Like these cells are similar, sorry, these spots are similar expressed spots. When it like when it isolate into U map, so it will keep as a group single place based on their expressions.
right. So let me follow up this. You mean you use the gene expression as a feature in order to do the graph-based clustering. So
Yeah.
that means in your complete data set, you have what? You have to uh, um, divide well the maximum expression. You find the interval of the maximum and the minimum expression level, and then equally divided that into 11 interval for, and then so that's why you have 0 to 10, 11 different cluster.
Yes, so that is absolutely correct. So based on,
Okay.
so it will first look into the what 1,000 genes in each spot is expressed. So first 1,000 genes. Zero. These 1,000 genes are like zero. So first is it has each spot has top marker, top expressed. For suppose top 100 or top 1,000, right? Then it went inside again. First we have spot level expression, so we know what are the top expressed markers based on their expression level. Then it will go to another spot and it will look into the another spot what are the top markers. Then like that it will go to the 1,000 spot. So then again these 1,000 spot, again it is making as a group. Okay, these 400, these 300. So these 300 means spot 1, spot 10 and spot 15. These are having similar markers, similar highly expressed markers genes. So it is assigned as a group, then that is cluster. So then it is given as 0, then again it will start, so from second one onwards, so again it is assigned as 1. So I have better picture, I think, to understand this. Just give me a second. From my single cell data, PPTs to learn materials. Okay. Okay, this is I'm just showing example from my single cell data analysis. Um, so initially it is it is having 0, like all samples considered as like one big group. But when it isolate top 1,000 genes, so these genes are different from these, and these like that. So it will isolate based on similar expression. So what kind of, like, for suppose 1,000 into like this is 300, this is 200, 100, 100, like that. It is split into spots. Then again dive into the again second layer, so we can see again within these 100, again there are some are having similar expression based on their like second layer. So like that, so it will in this scenario we can find. So yeah, so we can find what are the uh, like optimum number. For example, this is uh, yeah, resolution. So where exactly it stop splitting, like too many cells. If we see too many spots, sorry. So if we see when it came to 11th time or 12th time, I think this is a eighth time. Yeah. So eighth time, this is all are equal. So there are splitting into other groups, but they are not much changing. So this is the most standard one, 0.8. If we don't specify our analysis, let's go to, okay. Yeah. So here, this is a find cluster. We can also tune this one. Hmm. Just, yeah, resolution. So we can say like 0.8 resolution, that is giving us different let's see, um, I'll give you a small demo. So this is a resolution. So based on this, there is some algorithm. So based on this, it is like usually take 0.8. Let's take 1.5. Okay. Then it is giving us, you can see previously it is 14 different spots. Now it is increased to 15, 16, 17, 18, 19. So almost five more clusters it is observed. Like you can see, if I increase to 1.5, so it is splitting the um, uh, splitting the spots into five more groups like this. So this is like we always, before we are going to um, run the analysis, we are going to split what is the best number of split, like we can see what is the best number of resolution that we can see, like where from here onwards it is constant. So there is no change, so that this is we can consider as optimum. So these three. So even 0.7, yeah, almost these all are not changing much. So these are like going back to the down. So these are, for suppose I can say this is a CD4 cluster, so T cluster, T cells. Again, T cells, we have CD4 cells. So maybe, yeah, because of that markers, um, it is split into two, CD4, CD8. Again, CD8 cells, we have different markers. And again, CD4, we have different, like that. So we are diving into uh, different subgroups. So that is where like the clustering play key role. So in this here, that is single cell, same principle here. So we are diving into different clusters. So now uh, so now it is 10. So if I increase the more resolution, so I'll get more spots. Then if I have capacity to study more. But sometimes what happen if we don't get enough number of spots, so like it is very hard to validate that information. So my previous study, I have 500 plasma cells in one group, but it is very hard for us to validate those 500 cells. So that is always like if we have good validation data set, that is reveal that yeah, these cells are true. Then but in spatial information, you always understand what is the cell interactions here. So within the spot, like within spot 8, so there are two cells, they are interacting each other. So they are one is primary cell and another is secondary cell. Let me, I think I'm taking too much of your time today. um
It's okay. Yeah, I'm trying to understand. Yeah.
yeah, because spatial initially, uh, I thought it is screening of cell communication in spatial level. So I'm just referring to the one of the paper, screening of cell communications in spatial transcriptomic via collecting optimal transport. So here let's dive into the results. So multi-species ligand receptor. So this is one of the uh, yeah, ligand receptor interactions. Let's see, it's not okay. In CCI, they have better understanding. Okay, this is okay. Think so. This is how it looks like usually. Uh, this spot is moving towards the other, like this side. That means here there are two kinds of cells. One is epithelial and another one is something. So they are moving like based on their marker expression, they are saying that this is signaling. Like they look into all the neighbors and they will confirm like IGF signaling here. And similar way, spatial. Okay, this is they have like cell type information here. They have majority of the cells like primary cells, and they within the primary cells they have secondary cells spots. So that will reveal us the interaction between these two spots. Okay, these two spots. I think I'm looking for better. This paper is, I think, for Yeah, this is the methods again. Okay. Just looking into my like here's my spatial analysis. One of like my boss in Baylor. So he is working on spatial right now, spatial transcriptomic and all these. Um, I'm getting across. Yeah, they are published in abstracts. Compartments. This they use the okay graphical abstract three cells. Let me see if they have spatial information. This is a single cell RNA sequencing again. They have some nice papers on spatial. Okay. Okay, I think yeah, I'll explain this part yeah, like maybe next time uh, because that is most challenging part, that is integration. So now I think I have given basic idea what is spatial transcriptomic analysis. I think
Yeah. Okay. So can you go back to the tree diagram you showed just now?
yeah,
The one you start from
yes, yes.
one, say on the top level.
Hmm.
So you mean physically? It means in a two-dimensional space, there's a single circle on the top is the so-called one of the spots. This is what the definition.
So
This is a spot.
so in initially it considered these all are one. So now these all are cells spots, right?
Uh-huh.
So this is like zero. Everything is zero. Initially, every spot is zero.
So this is zero means a whole tissue region
Yes, yes, yes.
with
So
a okay. So
so
usually the size is about millimeter times millimeter
yes,
size.
yes, 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 yes. So they like that size part. I think so this like initial first step is it consider entire thing as zero. So all cells are all spots are equal. Then second step, so it look into the top highly variable genes. Like there is like it is a looping system. So it is try to find like there is a parameter like resolution. So if we want to see 8 times 0.8 times so it will run 0.8 times. Okay, this is the first we like when it runs the first time. So it observed these like these top spots. Then again, so again this top spot it take this and again it runs within the spot again. So what it splits the uh, two cell types. Okay, it is seeing there is a different markers. So like this like these 100 is expressed differently from these 100. So like that. So based on that it is like a tree. So same like it is splitting into like where it came to constant level, so until this there. Then from 0. So initially it is considered as 1, and now it is considered as 19 because I have given 1.5. Let's go back to 0.8. So that is 0.8 is optimum that is used by Surat package. So this parameter we always um, so you can see it is now came to 14, like 0 to like that is 15 clusters. So 15 spots are again within the 0 there may be two more spots or maybe four, there may be two more groups. So that is where like most of single cell and spatial analysis, they want to see, for example, even this 4, this is a CD4. So again, CD4 highly associated with some marker. So they report, yeah. So we found that this particular CD4. So again, we subdivided into CD4 like marker. They usually assign marker name plus CD4, then marker name plus CD4. So these cells are like these cells are showing similar pattern and similar uh, interactions, and we found this is a new cell types because this marker is highly associated with um, these cell types like that. Like that is, I think I can explain next time better. Right now I can we can just see another, for example, just okay. So when we see 0.1, this is a very low resolution. So it found one, two, three, four. Yeah, sorry, there is also two somewhere.
Uh-huh.
Okay, let's zoom in this. So it found four, five clusters. So five spots are these all are having similar expression. So again, we went down one more step. So again, these zero, how many are uh, like similarly explained? Maybe these group, these are like second layer. So like that, we are giving layer by layer, layer by layer. So again, zero, we are splitting into some other, one, we are splitting into some other based on their uh, like expression of markers.
Uh-huh.
So yeah, that is where we are looking into this. So usually optimum is we don't need to set up anything. 0.8 is a constant one. So uh, so that is that will give us the like nearly 15 markers. Uh, let's see, even if I don't want to specify this one. So you can see this is giving me given me 15 different group of spots. Yeah,
Uh-huh.
that is where we are. So again, in these spots, some are like you can clearly see some are it is group assigned as all these are epithelial cells, like all these are dominated by epithelial cells. These may be dominated by immune cells. So that is what it is fitting into further analysis.
Uh-huh. So the image on the right-hand side, so
Mm.
is it typically composed of 5,000 spots?
Uh, it is right now. It may not
Yeah.
have 5,000 spots, but typical image, big images, they have 5,000 spots. Like for suppose, um, let me
So
zoom
each
in.
little circle over there on the right image is denote
Yes, yes, that is
one of the 5,000 spots. Is that right?
yes, yes, yes. So
Mm.
here, I think. So my sample, just now I showed you. So this is one patient, like one tissue, right? One patient.
Uh-huh.
So he has like nearly 5,000 spots. So, but we can see there is like I don't have any tissue here.
Uh-huh.
So that means there are also spots, uh, but there is no tissue over there. So
Uh-huh.
so this square is 6.5 into 6.5 mm. So whenever we place this one on top of it, so yeah, whenever we place this, so if there is a tissue, it will capture. If there is no tissue, it won't capture.
Okay.
So
Uh-huh.
yeah, hardly we can see here. So uh, if this is whole 6.5 and 6.5, but we have only tissue this area, so it will capture this. So there is
Oh.
also bigger, there is also different types of capturing slides. So some slides they have bigger tissue, like big, maybe embryonic, embryonic tissues of mice. Those kinds are big, right? So it will have more spot, maybe their spot level more than 500, 5,000. They maybe 10,000 or more than that. So it is depends on analysis. Depends
I
on their,
see.
yeah. Now we have 5,000. So our spots are maybe less. What can I say? Maybe less than 5,000. I can say maybe 3,000 or 2,000 spots. We know that. We know that how many spots we have. Let me give that also so that. So let's, if I want to see, this is the information. Okay, so these are the spots, 2,696 spots I
Uh-huh.
have.
Okay.
So again, these within these, they are cells, different cells. So yeah, it is having all the, okay, let's view because so all the information is stored in our metadata. So this is kind of the information that is stored so this is single spot. So
Okay.
like this is a single spot. So this single spot have these many spatial counts, that is transcripts, and these many features like 4,242 genes, and these all are after this is after what I can say, like after normalization these are retrieved, and it is this entire spot is have considered as a one, like cluster one. So
Uh-huh.
okay, we can see the different resolution 1.5, 1.0. So we just used. So it will store all this. So again, so the deconvolution methods, so they will identify what is the proportion of cells present in this, like within this single spot. So there may be 2,000 markers highly associated with epithelial, and another 1,000 markers are highly associated with fibroblasts. So there it is considered, yeah, there are two cells in the spots. So these two out of 10, like there is a, uh, this is a slide one. So there is a, you can see 1, 1, 1, 1, 1. So these all are spot level. So
Okay.
there are many spots, slides. Yeah, this is a barcodes. Okay.
But this data set, you don't need the XY coordinate, the location, do you?
We do need XY coordinates because uh, right now I'm using publicly available data set.
Mm,
So from CIRAT,
I
as
see.
I mentioned. let me go back to the, so you can see here tissue position. Okay. So this will store all the information. This is a like bridge between the image and the features and all the information, physical information. So this will, I think one of the code, let's go back to the spot, just trying to,
Spatial feature plot.
yeah, yeah. Let's not this, brain one, okay. Okay. So you can see whenever I move the cursor, so you can see the coordinates in downs here. Okay. So now it is null. Let me, now it is null. So whenever I went to the one spot, so this is a spot, is their coordinates, x coordinate and y coordinate. So 1549.00 and 9032.00. So this is the
Oh,
spot information. So that is stored in our uh, mm, let me close this one. Yeah, that is stored in tissue position.csv file. So
see.
whenever so there are two, so these are all the information present in separately. So we need to upload these ones separately and we need to look into. But all the information is present in filter feature BC matrix H file. So all these three plus these two are present in single file format. So we can simply let me see my other. Okay. So this is my other code from my UAB. So if I want to run the like all the information, so this information contains all the barcodes and um, all these three barcodes, features, and matrix and position. So then like there is a spatial like spatial folder within the like this is a sample one. So each sample, uh, let's dive into that. Okay. I need to log in again. So again, within the spatial folder, we have the information of image. Uh, Let me see how it's. I think for one day, I think it is too much information. It may also confuse. Okay. So this is the space ranger, which is a pipeline that is getting like our information from counts. 10x genomics have that pipeline from counts to raw files, like FASTA files to these kind of files. P01. So this is one of the samples. Let's see. Okay, so this having all the information, filtered feature BC matrix, And let's see what kind of information we have. Okay, so the spatial. So here, keep this side by side.
So for a real case study, it can be achieved using your notebook. You don't need high-end GPU computing resources, do you?
If it is 10 G, for suppose my PC is 48 GB, like Mac. So for 10 samples, I don't think we need a big memory. But so the time I am saying like the only time it will take like a lot of memory is integration step. So uh, we are going like whenever we are going to like merge all these samples, so we are going to integrate based on their expression profile, top highly variable genes. So that is the time, like for suppose in my local computer it may take 12 hours, but in my right now my previous lab, it may take maybe 6 to 5 hours. It is depends on parameters also. uh,
In your previous lab, how long it takes?
For single cell data, it is taking depends on a memory and all 8 to 12 hours for me.
To, where that is it running on a GPU machine?
Yes, yes. So we have
Oh,
separate cluster. Sorry, we have separate HPC computers. So we always log into that HPC computer, and we can uh, we'll just run all these steps.
okay, I see. Oh,
So this is one of my recent struggling thing. So so this is single cell RNA. So I'm just integrating two data sets. So each have, this is one Smith data, and this is another Hankton data. So I'll immediately, like initially I just remove the less less, I can say, um yeah, small, like not important single cells. Then uh, then like these all are filtering techniques. So the time it, like this is a what it will take time. Now I think they are more updated methods. So this is a find integration anchors. So it will run the almost different number of combinations to see top 2000 genes in like in each cell level, then integrate all these cells into one group. So this step usually takes a lot of time. So anchoring means like connecting one cell with another cell based on their top markers, top features.
oh, okay.
Yeah,
Hey,
but
under the NCBIGO database, but have they include the spatial RNA dataset,
yes, there are many data sets available Geo. Um,
including the spatial
yes, yes,
RNA? Oh, okay.
yes. Let's see.
So one can look up those publicly available spatially RNA data.
Yeah.
Okay.
So they have some here. They are many data set. So triple negative breast cancer, correctly, okay, African American women. I think this is yeah, Copton J. So this is currently I'm using. So they have tar file. So within this, they have all the information. You can see. Let's see. This is 35.1 gigabyte. Um,
Okay.
let me stop.
There's a lot. Okay.
Yeah. So download aspects. Okay. So like
Okay.
that,
Okay.
we can download the data and we can use the data because this is most of the time it will take us to analyze only like step like if I use a local computer, so it may take little time. Like for suppose if HPC computers, if I'll take like 4 days, maybe here I may take 10 days. So, but like we can cut down these days because uh, there are many integrating methods right now.
I see. Okay.
Yeah, so there are right now I'm using spatial data integration analysis. So it did spatial analysis of spatial image based sequence based and VCM HD space dataset segments. So let's see. So whenever, okay, did they have integration here?
So it looks like Surat is the most popular package.
Yes, for R, Surat is most popular package. So integration of VCM HD spatial dataset. So we can practice like they have their already have the uh, so they have single cell and spatial, so we don't need to download the data, so we can for practice, so we can download their data from the like download like from yeah arrow package something. So they have some data, so yeah, new mouse brain, so we can download from here. So it is they will give us the very less memory so that we can even practice from our lab. So just normalization, visualization, and unsupervised clustering. So, and yeah, these all are the techniques, switch and like find variable features and scale data and all these. Then I'm looking
Okay.
for,
Good.
yeah, and these are the clusters that we are getting. If it is, this is single sample, I'm just trying to look into the special items and so this is they practiced on single you, we can see like they found the different regions and cell types, almost 29. So integration with a single cell, this is one of the deconvolution method that I am going to maybe next time I will introduce this
Good.
one. So there we can use the different methods, so based on marker gene sets. Yeah, this is a proportion of cell distribution of cell types across the like this is a cluster. So within the cluster, what kind of cell types? So in single cell, you will have only single cell type, but here you have different groups of cells. Yeah, s- sorry, proportion of cells and within the cluster and supervised cluster. Yeah, they are like they are much to understand in like down so that yeah, I'll explain this in next part.
Yeah. Okay. Good. Okay.
Yeah,
Ah.
let me stop
Good.
sharing.
Yes.
Yeah,
Yes.
I
Thank you so
think,
much.
oh my God,
Okay.
it is 2 hours. I think it is probably the
This,
late, right? 9 o'clock you from like in Taiwan it is almost 11.
yeah, that's okay, but is all right. Well, I learned something, right? I learned a lot.
Yeah, because it is a too big, so until and unless